Good morning. Good morning, students. Can you hear me clear? Come on. Okay. Is everything fine? Very good. Shall we begin? See the story. In last class, I think up to cell wall and plasma membrane we have discussed. Okay. Now, let me compare some amount of cell wall information and plasma membrane information. Then we will talk about the remaining story. First one, let us see cell membrane and cell wall. In this, cell membrane is majorly fluid in nature. Of course, you know, sometimes you call it as quasi fluid nature, sometimes you call it as semi fluid nature, etc., etc., but anyhow it's fluid. But when it comes to cell wall, generally it is considered as rigid. Cell wall is rigid. Even though the cell wall is rigid, it is capable to grow, which means especially primary cell wall, when you consider primary cell wall is capable to grow, but it is rigid. Keep it in the mind. Next one, when you talk about cell membrane, it is selectively permeable, or you can count it as semi-permeable. You know the difference. Semi-permeable means up to some part they will allow. Selectively permeable, only selected substances could be moved from one place to another place across the membrane. Next one, cell wall. Cell wall is permeable in nature. Permeable. If any material that tries to move away from the cell or it tries to come into the cell wall will never stop it. Cell wall will allow. Okay, you want to go? Go. I don't have any issue. As long as you are not damaging my structure, I don't have any issue if something is moving through me. That's the nature of cell wall. But plasma membrane is not like that. You are coming. Are you actually necessary? Okay. If you are not necessary, you should not go. That's a kind of nature called cell membrane. Next one. Fluid nature of the plasma membrane would be having some qualities like cell growth, cell division. These are developed due to fluid nature of the plasma membrane. Next one. When you talk about plasma membrane, it also has exocytosis nature. Exocytosis means something that is going away from the cell vomiting or cell secreting something, cell secretion, exocytosis. Some cases even it will have endocytosis nature. Endocytosis, which means endocytosis taking inside the cell. Some substances, they are coming into the cell. The incoming of the cell is known as endocytosis. You can take endocytosis as in the form of pinocytosis. Next one, phagocytosis. Pinocytosis or phagocytosis. What is pinocytosis? If a cell has intaken some liquid substances, that liquid intake is known as pinocytosis. If a cell has taken solid substances, that's called phagocytosis. In simple term, okay, if some kind of water engulfed pinocytosis, I mean liquid engulfed pinocytosis, some solid has been engulfed, that is known as phagocytosis. Next one, when you talk about Cell wall will not have all these things. It is not responsible for cell growth, cell division, endocytosis, exocytosis, etc. But one thing, cell membrane responsible for formation of intercellular junctions. Formation of intercellular junctions. What does it mean by intercellular junctions? Intercellular junctions are those cytoplasmic connections between the two neighboring cells. For example, you have two oil drops on the surface of any place. Two oil droplets we have. Make the two oil droplets come close to one another. If they come very close and touch, these two oil droplets will mix up or fuse and form a large droplet. This is possible only when the substance is fluid. Observe, I have two solid drops and I will make them colloid. Do they become one body? Never, because they are solid, individual parts. Because of the fluid, plasma membrane will have this property, which means I'll take one cell plasma membrane, which is fluid in part, another cell plasma membrane, which is also fluid, and make them to come close to one another. When they come close, then some places this plasma membrane lipid, that plasma membrane lipid will join, because lipids could be fusible. That joining will make the two neighboring cells will get connected in the form of small tube-like connections. Those tube-like connections, in case of animal cells, we call them as gap junctions. Gap junctions, variety of junctions are there. Or in plant cells, they are called plasmodesmata. Plasmodesmata. Like that, formation of intercellular junctions. But next one, if you talk about functions of cell wall, it will protect the cell. First of all, major work of the cell wall, protect the cell. When it comes to protection to the cell, it can protect the cell from infections. Any pathogen tries to enter inside the cell, cell wall will stop it. And also protect the cell from undesirable, means unwanted macromolecules. Any coming and hitting our cell, the cell will not have any issue if the cell has cell wall. Next one, when you are talking about cell wall, another property, okay, it will form or it will develop interaction between the cells. What do you mean by interaction between the cells? Imagine there is a plant tissue there. One cell connected to another cell. With the help of what material it will connect it? Do you remember? Intracellular cementing substance, that's called middle lamella. It's called middle lamella. Is it okay? Middle lamella. This middle lamella keeps the two neighboring cells stay together so that this cell can easily interact with another cell. In between the cells, if they wanted to interact, do they have mobile phones or any other kind of social media between them? No, not possible. Because these cells, if they want to interact, they need to stay together. That staying is possible with the help of middle lamella. Do you recall what was this chemical substance that is present in the middle lamella? Calcium or magnesium pectates. Most probably calcium pectates are present. Like that, cell wall make two cell interact. Cell membrane will make the two cells develop connections. Got the idea? This is about what we need to comparatively learn between cell membrane and cell wall. Then go to inside the cell. There is an area called cytoplasm. You know the cytoplasm, which contain cytosol and cell organelles. I have given in earlier class about this cytoplasmic division. Now directly I am coming to cell organelles. Cell organelles. Cell organelles, a kind of network of membrane and non-membrane bound structures which perform various functions required for a cell to survive. Cell organelles, organs, bodies, organelles, small parts. Okay. Organelles, let us check. Some organelles are double membranous. Double membranous. Some organelles are single membranous. Single membranous. And some are non-membranous. Non-membranous. Among them, the major part is single membranous. Non-membranous less number. Double membranous is also less number. First one, mitochondria, chloroplast, and nucleus. These are all are double membranous bodies. Nucleus is also possess two membranes. If you decide nucleus as a cell organelle, you have to mention it's a kind of double membranous cell organelle. What about non-membranous? Ribosomes. Then another one is called centrosome. Of course, there are two centrioles which are form this centrosome. Now my discussion is single membranous bodies. Apart from this, whoever present, obviously they become single membranous. In that case, when you talk about single membranous cell organelles, immediately you will get one idea. Some single membranous organelles are called endomembrane system. Endomembrane system. Some single membranous bodies are called endomembrane system. What does it mean by endomembrane system? What you talk about endomembrane system here? Imagine if I say some cell organelles are considered as endomembrane system. There, first thing you should remember, their functions are coordinated. Their functions are what? Coordinated. What does it mean by functions are coordinated? Careful. Whenever I say A, B, C functionally coordinated, I am saying in this sense, A complete one work, send to B, B complete some work, send to C, like that A, B, C work together to complete the process of any kind of action. That's why it is called coordinated action. In simple terms, work together. Whenever they are work together, their functions are coordinated. What are those cell organelles whose functions coordinated or who are working together? In that, first endoplasmic reticulum, next one Golgi complex and lysosomes, and finally vacuoles. These are the cell organelles which are considered as endomembrane system. What does it mean by this? Whenever you talk about Golgi complex, endoplasmic reticulum, lysosomes, vacuoles, all of them present very close in association because they work together in order to complete variety of works. If a cell has no endoplasmic reticulum, that cell obviously does not possess Golgi complex. If a cell has no Golgi complex, it cannot develop lysosomes, vacuoles, because they develop like this. Endoplasmic reticulum will produce Golgi complex. Golgi complex will develop lysosomes and vacuoles. If a work somehow completed by endoplasmic reticulum, later that work will be allotted to Golgi complex. Then the Golgi complex will decide where this further assignment of work or where the work can be assigned further. This kind of thing is called endomembrane system. But when it comes to single membranous bodies, some single membranous bodies are also mentioned as non endomembrane bodies. Non endomembrane system, or there are some single membranous who can work independently. Best example, there are some cell organelles called microbodies. In that peroxisomes, next one glyoxysomes, etc. There are few other more are there. They have single membranous system, but they do not work along with these bodies. Peroxisomes work independently. In fact, peroxisomes have very few functions. Among them, peroxisomes responsible for removal of hydrogen peroxide from the cell by any metabolic pathway. If H2O2 has been synthesized in the cell, that H2O2 would be eliminated with the action of a cell organelle called peroxisome. Peroxides are very dangerous. That's why cell has separate kind of organelle which can eliminate peroxides. Among them, peroxisomes will do that. Anyhow, observe. It doesn't matter whether it's a single membranous or a double membranous. If any cell organelle could work independently, that would be considered as non-endomembrane body. If it work in association or in coordination with other cell organelle, that should be considered as endomembrane body. For example, tell me, what about mitochondria? Is it an endomembrane body? First of all, is it a membranous body? Mitochondria. Is it a membranous body? Of course, mitochondria is a membranous. In fact, it has two membranes. But problem is, mitochondria would work independently of other membranes. It can work by itself. Chloroplast also can work by itself. Nucleus also can work by itself. It doesn't matter remaining working or not. They can all work by themselves. Those bodies which are working by themselves are known as non-endomembrane bodies, which are working only in coordination with other, are called endomembrane bodies. Is this words clear to you? That's why first we will discuss about endomembrane body. In that, endoplasmic reticulum is the first choice. Now, ER or endoplasmic reticulum. ER or endoplasmic reticulum. In electron microscope, this is an interesting thing. In electron microscope, what does it mean by that? If you wanted to see endoplasmic reticulum, you need to have electron microscope. There are some organelles easily visible under the simple microscope even. There are some organelles, if you want to see them, you have to stain. That stain will make them to be visualized. But there are some organelles, they will never be visible even with simple structures. Among them, endoplasmic reticulum is one. You need to apply the extraordinary electron microscope to see endoplasmic reticulum. Under electron microscope, it appears It appears as tiny tubular network. Tiny tubular network. In electron microscope, it appears like a tubular network. Since it is forming a network-like structure, that's why they have given a title called as endoplasmic reticulum. Reticulum means net-like structure, not internet-like. Net, mesh-like structure. Okay. They have tube-like arrangement of the membrane, so that membrane which is tubular arranged, this is known as reticulum or net-like structure. Next one, I'm going to take how does it exactly appears inside a eukaryotic cell. Consider this is a eukaryotic cell. It's a cell. It has the main structure that's called nucleus. I'm taking a small nucleus here, but don't think nucleus will be smaller than remaining things. But in requirement, I have chosen here a small nucleus. Okay, this is the nucleus. You know, nucleus is a double membranous body. It has outer membrane and inner membrane, and it has structures called chromosomes or chromatin. Excellent. The outer membrane and inner membrane will be separately present. This is the nuclear body. Then the outer membrane started to expand. It started to expand like a tiny tubular structures all over the cytoplasm of a cell. This tiny tubular structure, you have to call it as endoplasmic reticulum. Endoplasmic reticulum. Almost entire cytoplasmic area would be occupied by this endoplasmic, almost all corners, this endoplasmic reticulum will go. If you see the endoplasmic reticulum appearance, this is the cytoplasmic space. Now, the endoplasmic reticulum will divide the cytoplasm into two places. To understand it better, take a balloon, blow it, keep it in a bucket and pour water. Don't make the balloon to come up. Keep the balloon inside. Now in the bucket, you have two places now. Two places. One is inside the balloon, that empty space. Outside the balloon, watery place. Like that, I have a cell. Inside the cell, I have kept a tiny balloon-like extensions. Those are called endoplasmic reticulum. Now this endoplasmic reticulum would divide cytoplasm into two places. One place is called lumen or luminal space. Luminal space is nothing but the space inside the endoplasmic reticulum. If I come outside the endoplasmic reticulum, between the cell membrane and the endoplasmic reticulum, you would see extra luminal space. Extra luminal space. That extra luminal space is nothing but cytoplasm. Extra luminal space is cytoplasm. The statement would be like this. Endoplasmic reticulum divides cytoplasm or cell space into two environments. One is luminal space inside the endoplasmic reticulum, extraluminal space outside the endoplasmic reticulum, which is nothing but cytoplasmic area. Now, because of endoplasmic reticulum occupy most of the area of the cell, that's why endoplasmic reticulum is also sometimes called as the endoskeleton of a cell. Inside the cell, skeleton, I will talk about this word later. They have skeletal body, that means tiny small rods will be present. These rods are nothing but microtubule, microfilament, intermediate filaments, etc. But along with all those rods, there will be a presence of the membrane. That membrane is nothing but endoplasmic reticulum. That endoplasmic reticulum will make endoskeleton of a cell. Okay, done. Come back to this membrane known as endoplasmic reticulum. If they are talking about endoplasmic reticulum, they do not talk about the space present inside the endoplasmic reticulum. They will talk about what is happening on the top of the membrane. That's called endoplasmic reticulum membrane. Now go to on the endoplasmic reticular membrane. Some places you will see there will be a presence of ribosomes on the top of endoplasmic reticulum. The ribosomes present area commonly denoted as rough endoplasmic reticulum, RER. If the place has no endoplasmic reticulum, we will call it as SCR, smooth endoplasmic reticulum. RER, SCR they were not present very distinctively. Just I have divided the endoplasmic reticulum into two parts, smooth endoplasmic reticulum and rough endoplasmic reticulum, only by the presence and absence of the ribosomes on the top of them. If ribosomes present, rough endoplasmic reticulum. Ribosomes are absent, you can call it as smooth endoplasmic reticulum. Getting the statement? That's the reason why we have two types of endoplasmic reticulum. But it doesn't mean that a cell will completely have smooth endoplasmic reticulum. A cell will have completely rough endoplasmic reticulum. That's not the story. A cell, on an average, rough endoplasmic reticulum, smooth endoplasmic, both are present. But if a cell has more requirement of rough endoplasmic reticulum, more accumulation of ribosomes on the top of it. That's it. Then come back to the properties of RER and SCR. Rough endoplasmic reticulum. Rough endoplasmic reticulum, 80S ribosomes present on the membrane. On the membrane, what type of ribosomes are present? 80S. Be very careful. You have been saying if ribosomes present, rough endoplasmic reticulum, ribosomes absent, smooth endoplasmic reticulum. Excellent, extraordinary. But what type of ribosomes they are? Are they smooth? Are they 70S or 80S? Always, if a ribosome get attached to the endoplasmic reticulum, that ribosome definitely should be 80S ribosome. Eukaryotes may have 70S ribosomes, but that 70S ribosomes always present in matrix of mitochondria and stroma of chloroplast. Apart from these two places, you will never find the 70S ribosomes in the free state or in the combined state on the cytoplasm. Okay, so RER will have ribosomes. What type of ribosomes? 80S type of ribosomes. Next one. In order to attach ribosomes on the surface of endoplasmic reticulum, they have some special type of attachment of ribosomes called ribophorins. One thing. You were saying some area of the endoplasmic reticulum ribosomes attach, some area of the endoplasmic reticulum ribosomes are absent. Why? The area where ribosomes attach, there is an existence of a transmembrane protein known as ribophorin. The ribophorins will trap ribosomes and make the ribosomes to get attached on the top of endoplasmic reticulum. Got it? Next one. If ribosomes are present, main work is protein synthesis. Rough endoplasmic reticulum main work is protein synthesis and also secretion of them. Protein synthesis and secretion. Synthesized proteins Rough endoplasmic reticulum doesn't like to keep it for itself. It will send them to, okay, another place. That sending process, they are calling it as secretion. Protein synthesis as well as secretion of those proteins away from the place, both the works would be done by rough endoplasmic reticulum. The cells which are actively involved in protein synthesis, they contain high quantity of rough endoplasmic reticulum. Best example, in the pancreas, beta islets constantly synthesizes insulin hormone, which is a protein. So, in order to maintain the protein structure, protein to synthesize excessively, they have some structure called rough endoplasmic reticulum. Then go to SER, smooth endoplasmic reticulum. Smooth endoplasmic reticulum, it is smooth because ribosomes are absent on the surface, obviously. Then ribosomes are absent. If ribosomes are absent, it cannot participate in protein synthesis. Then, when it comes to ribosomes are absent, it would be responsible for lipid biosynthesis. Lipid biosynthesis and also steroidal hormone biosynthesis. Steroidal hormone biosynthesis. What does it mean by that? Listen, plasma membrane having one property that's called self-repairability. What does it mean by self-repair? Somewhere else, plasma membrane has some small kind of tearing, or a small amount of plasma membrane has been damaged. That makes what? That plasma membrane should be repairable. Who will repair plasma membrane? Our smooth endoplasmic reticulum. It will send one vesicle and fill the gap. Like that, lipid biosynthesis, whatever the lipids that are required, all of them almost synthesized in smooth endoplasmic reticulum. There are steroidal hormone biosynthesis. For example, testosterone, progesterone, estrogen, etc. These steroidal hormones would be synthesized on the top of smooth endoplasmic reticulum. Be careful, in plants there are no steroidal hormones. As you know, plant hormones are auxins, gibberellins, cytokinins, and also ethylene, and whatever the plant hormone is there, it is never made up of steroid. That's why in plants, steroidal hormone may not be happen in plant. With respect to plant I am talking. Plant steroidal hormone will not be occur, okay? But when it comes to animal cells, there is a chance for the synthesis of steroidal hormone. One more thing. Next one. Detoxification of drugs and toxins. Any toxins or drugs entered in our body. Don't think drug in the sense always morphine, cocaine, codeine, etc. drugs are they are going to have some kind of stimulation. Don't think drugs are always like that. Drugs are any chemical that can be absorbed by our body and started to respond to that chemical. Simple. Paracetamol is a drug. When do you take paracetamol? Paracetamol is an antipyretic, which means it reduces our body temperature. In order to reduce body temperature, it has to work on hypothalamus. That means if this chemical is working on our hypothalamus, it's a variety of drug. Analgesics, antipyretics, variety of things which are working on our body, you have to call them as drugs. One thing, any drug toxin that entered in our body, first it has to reach liver, where smooth endoplasmic reticulum will be present. It has to processed by the smooth endoplasmic reticulum. In the liver, hepatocyte, smooth endoplasmic reticulum, they have to be processed. In order to process drugs and toxin, smooth endoplasmic reticulum contain some kind of protein called as P450 protein. This is present on our smooth endoplasmic reticulum, responsible for detoxification of drugs, any kind of toxins that are entered in our body. These are the functions of rough endoplasmic reticulum. These are the functions of smooth endoplasmic reticulum. This much of information enough. If you see the books present in the market, especially commercially sold books, commercial, do you understand what I mean commercially sold? Which means some books just printed and sent. There you will have a lot of information. Don't mug up all those informations because they may not make you remember the things properly. Stick on to the information that is given in NCERT book. In order to understand NCERT information, whatever the information that you need to study from different sources. But don't try to read all books. Those books, end of the day, will not be available with you and you could not able to study all of them. So stick on to NCERT content. Then come back to next endomembrane body, that's called Golgi complex. Golgi complex, Golgi apparatus, Golgi body, like that. Many names are there. Whatever it is, they are talking about same. Sometimes they call Golgi complex. Sometimes that is called Golgi body. Sometimes called Golgi apparatus. Okay, whatever it is. The name was given because there was a scientist called Camillo Golgi. Camillo Golgi or Golgi. He was actually a doctor, a neurosurgeon. He was working with nerve cells, especially a bird, owl. Owl nerve cell he had taken and he wanted to study about it. On the nerve cell, okay, on the nerve cell, there he added some chemical. That chemical known as potassium bicarbonate. He added some chemical, that means some dye. Upon addition of the dye or stain, some areas of nerve cell cytoplasm have been started to appear like a bright colored bodies, bright, darkly stained bodies he started to notice. Darkly stained ones, bodies in cytoplasm he had noticed. That darkly stained bodies are known as Golgi. Later those darkly stained bodies, they were called as Golgi complex after his name. Golgi complex or Golgi body and so on. That's a name development. Then where it is present and how it will be work. The working will be like this. Whenever we talk about Golgi body, it is present around the nucleus in the form of concentric rings. What does it mean by concentric rings? Listen, whenever you see Golgi complex actually formed by endoplasmic reticulum. Endoplasmic reticulum is the one which forms Golgi complex. Okay, but endoplasmic reticulum itself has come from outer membrane of nucleus. In that case, the outer membrane of nucleus is responsible for the formation of Golgi complex indirectly. Directly it does not come from the outer membrane. First endoplasmic reticulum come. After that there is a development of Golgi complex. If so, always Golgi complex, wherever it is present, develop around the nucleus in the form of concentric rings. Concentric rings. Next one. Golgi complex formed by endoplasmic reticulum. Keep it in mind. Formed by endoplasmic reticulum. Okay. How does it appear in structure? Which means I wanted to study structure of Golgi complex. In structure, how does it appear? Its appearance would be like this. For example, consider this one is endoplasmic reticulum. Not this much, but for our convenience, I am taking it is endoplasmic reticulum. You have an idea how endoplasmic reticulum looks like. This is endoplasmic reticulum. Around the endoplasmic reticulum, Golgi complex will develop like small tubes which are arranged like a stockpiles. These rings arrangement has a special name called cisternae. What do you call them? Cisternae. This is stockpiles. That means a flat tube-like structures are there. One tube will be arranged on another tube. All these tubes collectively known as cisternae. Cisternae. The stockpiles of lamellae or the stockpiles of tiny tubular structures are known as cisternae. Now, if you take this Golgi complex cisternae, one tube arrangement you are talking. For example, one tube. In this, it has some diameter. The diameter will be around 0.5 to 1 micrometer in diameter. One tiny tube. Many tiny tubes. One will be on the top of another. That arrangement I am calling it as cisternae. Very good. In one tube, if you check, what are the components are present? The one tube diameter will be around 0.5 to 1 micrometer in diameter. One tube. Like that, many tubes will be present. Interestingly, number of cisternae varies from cell to cell. What does it mean by that? Here I have taken 4, right? 4 tubes are there. Sometimes maybe 8, sometime up to it can go 60 tiny tubular structures will be stockpiled one on the top of another. That much length could be developed. Got the idea? Cisternae is nothing but stockpiles of tiny tubular arrangement. Each tube will be having 0.5 to 1 micrometer in diameter. Like that, if they are stockpiling, the number of stockpiles will be vary. Okay, somebody will have 2 also, somebody will have 4, like that. 2 to 60 tiny tubular arrangements could be there, but on an average, generally on an average, there will be an existence of 4 to 8 tubular structure. That's why I have taken 4. Then, how does endoplasmic reticulum form our Golgi complex? You said Golgi complex is formed by endoplasmic reticulum. How? It will form like this. If something the endoplasmic reticulum want to send, for example, this material, endoplasmic reticulum wants to send to our Golgi complex. It will send in the form of small vesicle. The material to be packed will be sent in the form of small vesicle. This vesicle will come along with these proteins or protein, any kind of material. Okay, it is coming in the form of small vesicles. The materials to be packed or processed in the Golgi complex are coming from endoplasmic reticulum. Now while they are coming, they come in the form of vesicle. This vesicle will come and fuse with the membrane of our Golgi complex, will come and fuse. The fused one, this is the same material. Okay, the fused one will releases this substance inside the cell, inside the Golgi complex. This is the one which has fused. Once it is released there, that material which is needed to be modified, this is the matter which is going to be modified by Golgi complex. Actually that was given by our endoplasmic reticulum in the form of a vesicle. That vesicle travel and come and fuse. That fused one releases that matter inside the in Golgi complex cisternae. This Golgi complex cisternae will take this matter and then started to modify, gradually modify and transferred from one cisternae to another cisternae, and finally it will reach us to one area, modified and gradually travel. And here it will start, comes out of Golgi complex in the form of small vesicle. It will travel into this vesicle. Okay, and gradually around that matter a small vesicular body will be developed. This is called secretory vesicle. Secretory vesicle. Are you getting what I am doing here? Simple. There is a material, this endoplasmic reticulum wants to send to Golgi complex. What does sending process? It is in the form of vesicle. I am endoplasmic reticulum, I am forming you, so I am throwing some matter, you catch it. It's not the thing. It will release a small kind of vesicle. That vesicle freely floating in the cytoplasm and come and mix with Golgi complex. Within the Golgi complex there is a lot of cisternae. These cisternae are interconnected and the matter will be started to move from one cisternae to another cisternae. While that matter moving from one cisternae to another cisternae, gradually it started to modify. So that while modification is going on, once final modification is over, that matter will be sent away from Golgi complex from this area. Now, with respect to this kind of modifications, you have on the Golgi complex two phases. One phase is called cis phase. Cis phase. Tell me, where is cis phase? Towards the endoplasmic reticulum or towards the away from the Golgi complex? It is always towards endoplasmic reticulum. Okay, cis phase. Since the cis phase present towards the endoplasmic reticulum and responsible for forming of endoplasmic reticulum, that's why it is called forming phase. It is called forming phase. Next, away from the endoplasmic reticulum, you will see the development of trans phase. Trans phase. Trans phase is present away from the endoplasmic reticulum. This trans phase is the phase where this Golgi complex contain matured proteins. The proteins which were fused in the cis phase gradually travel from one cisternae to another cisternae and they have come here. What type of proteins do you expect in the trans phase? Matured, properly formed protein. Is it okay? That's why trans phase is known as maturation phase or matured phase. In addition to these names, actually cis phase is in the form of convex. You can see that convex. Then trans phase is appear in the form of concave. A small kind of cave like structure is present. Is it okay? Upper surface is convex upper one, and the lower surface is known as concave body. So altogether cis phase is also called forming phase or concave phase. Trans phase is also known as maturation phase or convex phase. Interestingly, cis phase and trans phase are interconnected with cisternae. Cisternae are totally interconnected. Cis and trans phases are interconnected. Cis and trans phases are interconnected but entirely different. This is the interesting thing about them. They are entirely different. Cis and trans phases work is totally different, but they are always interconnected, because one cisternae will get connected to another cisternae. So always cis and trans phases are interconnected. If they are interconnected, it doesn't mean that they are having all functions are same. Cis phase function, trans phase functions are totally different. Understand? This is about orientation of Golgi complex. Cis orientation, trans orientation. Am I clear? Any more confusions and conflicts are generated to you? Just post it there. I could be able to answer. Okay, then after that, biological functions of Golgi complex. What are the functions the Golgi complex will do? If a cell has an organelle, obviously that organelles must be having a work. Out of decoration, a cell could not develop any organelle. Golgi complex, the big body, it must be having some work. So first one, the main function the cell will assign to Golgi complex is packaging and secretion. Any material could be moving away from the cell, that should be properly packed. Very clear, this word is this. If a material that is going away from the cell, that material must be properly packed and given to other. It's not that I don't want this, you take it, throw it. No. Packaged. This package is also having very significance. The package will indicate where this body has to go, what type of function this body is going to do. Everything will be written on that package. Very interesting. See, imagine beta islets of Langerhans synthesized insulin hormone. That insulin hormone has some work. Where this insulin hormone has to work and how this insulin hormone has to work, everything will be written in that package. Understand? That package will be carefully go and work, and then that packaging was done by our Golgi complex, packaging and secretion. Next one, another thing, glycosylation of proteins and lipids. What does it mean by glycosylation of proteins and lipids? Glyco means what? Did I give this term in earlier classes? Glyco means what? Glyco means carbohydrates. Sometimes carbohydrates added to protein. Carbohydrates will be added to protein. When carbohydrates are added to protein, that protein become glycoprotein. When carbohydrates are added, the protein become glycoprotein. Glycoprotein. A protein, nature is protein, but on the top of the protein, some amount of carbohydrate has been attached. Such kind of proteins are known as glycoproteins. Glycoproteins are synthesized by Golgi complex. Next one. Carbohydrates sometimes added to lipids. Then there will be a synthesis of glycolipids. Lipids do have carbohydrate moiety attached. That's called glycolipids. Either you take glycolipid synthesis or you take glycoprotein synthesis. Both would be done by Golgi complex. Now this word has a lot of conflict. Listen, the work of glycosylation either can be done by endoplasmic reticulum or it can be done by Golgi complex. Both can do. I am saying this word to you very clearly. Both Golgi complex and endoplasmic reticulum can synthesize glycoproteins, glycolipids. But in our NCERT book, they have assigned this work to Golgi complex. In here and there, you will read there will be a presence of glycosylation in endoplasmic reticulum also. But keep it in mind, with respect to NCERT book, we will follow that. That is glycosylation and glycoprotein will be synthesized by Golgi complex. Next one. Cell wall biosynthesis. Cell wall biosynthesis. Wherever but there is a presence of cell wall, eukaryote I am talking about, okay? Cell wall biosynthesis. Wherever you come across cell wall biosynthesis in eukaryotic cells, that cell wall is synthesized by our Golgi complex. Listen one thing. When it comes to the cells have cell wall, cells will not have cell wall. What do you think more in quantity? I mean in the nature, in the biological world I am talking. Some cells will have cell wall, some cells do not. What kind of cells will be more in quantity in our biological nature? Obviously, cells which are having cell wall are more in quantity. In fact, bacteria have cell wall, fungi have cell wall, plant will have cell wall, most of the protista will also having cell wall. Very few members do not have cell wall. One is protozoan, another one is slime mold. I have given gap because you have to recall it. Slime molds also do not have cell wall, and also animal cell, obviously. There, cell wall biosynthesis by our Golgi complex. Next one. Acrosome synthesis. Acrosome synthesis. What does it mean by acrosome biosynthesis? Do you remember? On the top of a sperm cell, there might be a presence of a small cap-like structure. That cap-like structure contains some enzymes in the name of hyaluronidase. This hyaluronic acid digesting enzyme responsible for breakdown of egg outer membrane. That's called zona pellucida. You will get that later. Acrosome present on the sperm cell head. That's why this acrosome is also synthesized by our Golgi complex. These are the major functions. Here and there some extra functions here and there you will identify. Okay, got it? Any more requirements? You can post it there. Today I wanted to clarify one thing. You will have exam up to endomembrane introduction only. Golgi complex, endoplasmic reticulum are not there for the coming Sunday exam. Okay, keep it in mind. Then any more requirements, any need of or else suggestions or any other kind of things you can give, I can accept, or if you want you ask me, I can also give. Come on, anything else? Are you following my class, my dears? Is it okay? Am I comfortable? Up to vacuole is not there. Okay, up to vacuole is not there. Come on, are you comfortable? The speed, the thing, what I'm doing? If you have any suggestions you can post, or any requirement in fact. Okay, we are done with today. We will meet in the next class. Thank you.